Thank you for inviting me. I've had a great day so far. Hopefully I don't lose you all in this talk. If there's any point in time where I'm not clear about something or if there's something confusing, please just interrupt and we'll have it be more conversational if that works. So I'm going to talk to you today sort of in sort of a mishmash of sort of more overview, sort of conceptual overview of what I mean when I say the word epigenetics and what does chromatin mean, just to get you up to speed. And then I'm going to present at least one, maybe two pieces of data from my own lab, which is still a very young lab, that gets at sort of information flow in epigenetics. And so how is there something heritable beyond the Watson-Crick base pairing capacity of DNA? And so we're trying to understand the molecular players here. And in the context of this incredible compaction problem, it's sort of the existential crisis, if you will, of every single cell in your body. How do you fit 6 feet of DNA in a 6-micron nucleus? It's approximately a 6 order of magnitude compaction problem. So how does this compaction get achieved, right? How do you jam? It's essentially the sort of problem is like fitting a garden hose the length of the Mississippi River on this stage, okay? Just to give you a sense of how much length of DNA we have to compact into a very small space. So it's a problem of compaction, and it's also a problem of meaningful compaction. So arguably what defines a given cell type is its chromatin state, which genes are on, which genes are off. And so how you squish that DNA into the nucleus in such a way that you keep certain genes on and keep certain genes off is the essence of the problem. And so this has some interesting connections. How this packaging occurs has some interesting connections to heritable information systems that exist beyond the double helix. Okay, so where should I stand? Can I just wander around? Am I blocking anyone? Okay. So there's this interesting phenomenon. Well, actually, epigenetics sort of historically has been very much a catch-all for things that we can't explain with genetics. And it's been sort of a murky backwaters of science for many years until we started to understand some of the mechanisms. And it's still a very young field, so I'm going to be saying this a lot, but we don't understand all the details still. And that makes it exciting because it's an active area of research. And so here's sort of an interesting way of presenting this problem. You can have 2 genetically identical twins, but the concordance rate for major depression, that is to say, the likelihood of one having major depression, of a second twin having major depression if the first one has major depression, is only about 50%. So that argues that even if they're raised in the same household or in different households, that's controlled for. So that makes the argument that major depression or other sort of complex psychiatric diseases are, only at best half genetic in basis, bringing up this sort of age-old question of nature versus nurture. And what is the molecular basis of nurture is maybe a better way of posing the question. So how does the environment, how do inputs from the environment sort of molecularly transduce into stable phenotypes that exist in the lifespan of an organism and can actually be heritable, not in a DNA sense, but in an epigenetic sense to the next generation, okay? So I want to challenge this widespread assumption in modern biomedicine that genes determine disease. I think this is largely true, but there are many examples, particularly complex psychiatric disorders, that don't have a genetic basis. People have really been looking for genome-wide associations for various markers, and the claim is that perhaps major depression is a constellation of different gene susceptibilities. Or the other explanation is that it has nothing to do with genetics and is entirely epigenetic. I don't think we know the answer yet, but I think there are instances where genes do not determine disease, even diseases that are inherited. And so I'm going to present a little bit. The first part of the talk is going to be really at the molecular level, what we know about chromatin and epigenetics. And then the last half of the talk is going to be sort of centered around sort of a few sort of cherry-picked examples of epigenetics in disease. And it's by no means a complete set. It's a very small sampling. And there are a number of very interesting cases that I had to leave out for the purposes of this talk since I don't have 8 or 9 hours, right? Okay, so getting back to the central dogma. So this is sort of the essence of molecular biology, right? That DNA is sort of the heritable information carrier, right? So information storage polymer, And then protein is sort of the functional end of comprising all the enzymatic and sort of functional properties of cellular machinery. And then mRNA is just sort of a boring intermediary, okay? So the central question, absent any sort of notional epigenetic inheritance, is that this is where all of the information lies, in the Watson-Crick base pairing capacity of DNA. The ability of DNA to pair in a polymerase with an incipient RNA species that grows to full-length RNA as a consequence of polymerase activity. So this pairing, this Watson-Crick base pairing, is sort of at the essence of this transcription process, right? And so the ability to form Watson-Crick base pairs, this beautiful double helix, which we're going to investigate a little bit more closely in a moment, is sort of the essence of this information decoding process on its way to being functional. Okay, so the question I want to pose today is, does nature keep more than one set of books? Is this DNA the only information carrier that's heritable? And are there other information carriers that exist? And the answer is certainly yes. We don't fully understand exactly the molecular details of all of these, but there are epigenetic information carriers, some of them well established, some of them still somewhat on the path to being established as epigenetic information carriers, putative epigenetic information carriers, if you will. And then some sort of decoding system, some sort of reader system that reads this epigenetic information and somehow transduces that information into a consequence, much analogous to sort of DNA in storing information, transducing that information into protein function. Okay. So just one more bit of sort of verbiage to get out of the way. This concept of epigenetics. I'm going to give you my operating definition in the sense it's not, strictly speaking, what everyone would call epigenetics. I think the difference in various people's definitions of epigenetics is this concept of heritability. So I define epigenetics as a heritable, mitotically or meiotically heritable phenotype that is not due to a change in the Watson-Crick DNA base pairing capacity, and is often traceable to sort of stable expressions in gene expression, somehow encoded in the way DNA is packaged, i.e. chromatin. Okay? So when I say chromatin, I mean just the way DNA is packaged, and we'll get into more detail about that. First, just sort of a primer on how one views these things, because I'm going to be showing you a few molecular structures throughout the talk, because it's the level of resolution that we have for understanding some of these processes, these decoding processes. And so I just wanted to show you just a couple different ways of rendering DNA, if you haven't already seen this, or a molecule. So this is a crystal structure of DNA that's rendered in several different ways. That was a surface representation. This is sort of a cartoon ribbon representation. This is more of an atomic coordinate representation. So they all represent the same atomic coordinates. We know every atom's position. And it's just a way of how do you show this simply to the human eye? And so this is a surface rendering on top of the atomic coordinate position. And then here's a surface again. So as we look at structures, I'll show you sort of a variety of different ways of rendering them. And they sort of help us understand something. Essentially, if you know the structure of something, you know a lot about its properties. You can tell a lot about how it functions. Simply deducing the structure of DNA from a few small parameters enabled Watson and Crick to devise the central sort of dogma in molecular biology that this double-stranded duplex of DNA could code for RNA and could template itself for replication. Okay. So that's a great example of knowing a structure, sort of giving you a lot more information than just knowing the molecular composition. Okay, and so explicitly, a lot of what I'm talking about today is occurring in the context of transcription factor-driven processes. So I would be remiss if I didn't upfront say a lot of what we're talking about in terms of epigenetic processes are dictated or driven by sequence-specific DNA binding factor events, okay? And so a lot of what happens to the genome is governed by sort of binding events at the DNA level that are sequence-specific for the DNA that involve some sort of recruitment process, either a polymerase or histone modifying activities. Conversely, you can have sort of a gene repressive activity that perhaps competitively binds the same sequence on DNA. And so it's the concentrations of these factors, the activator versus the repressor, and their actual affinities for the sites that dictates whether a given gene is on or off, simplistically. So this sort of framework for understanding typically more prokaryotic types of systems is useful for thinking about epigenetics as well. Typically, we're talking about chromatin acting as a repressor of some sort of transcription, sort of a baseline repressor, as opposed to needing an actual repressor. Okay, so you're all hopefully familiar with this year's Nobel Prize for medicine, which was given out for induced stem cell pluripotency. This is just sort of the sort of general concept of embryonic stem cells, where you can take an embryonic stem cell and differentiate it down many different lineages by putting it in different cocktails of factors. You can also culture embryonic stem cells indefinitely because they have this capacity for self-renewal. So you can take a pluripotent embryonic stem cell, one that's capable of creating any sort of tissue, and differentiate it to precursor cells which are multipotent that can maybe that can give rise to a restricted set of tissues, and then you get down on one end to sort of more terminally differentiated tissues like neurons, muscle cells, kidney cells, etc. Okay, so this is sort of the general differentiation paradigm, and in each of these cases, there's a chromatin level change. Both transcription factor sets of controlling gene expression change at each of these points, but also chromatin level changes occur. So the Yamanaka work really was given for adding this cocktail of 4 transcription factors to a terminally differentiated cell type, in this case skin fibroblasts, although other fibroblasts work. And this cocktail of 4 transcription factors is sufficient to de-differentiate these terminally differentiated cells back to something that resembles very strongly an embryonic stem cell, otherwise indistinguishable from an embryonic stem cell. Okay, so this is a fascinating result, but it's extremely inefficient. About 0.05% to down to 0.01% of the cells that get all 4 of these factors are actually capable of de-differentiating. So why is that? Also, it's cell type dependent. You can't take a neuron and give it this cocktail of 4 transcription factors and turn it back into something that looks like an embryonic stem cell. So why is that? It turns out that there's a barrier of some sort to reprogramming that makes it very inefficient. But it's not a complete barrier. It's just a very high barrier. Some small percentage of the cells that get these 4 transcription factors are able to surmount it and de-differentiate. So what is this barrier that makes de-differentiation so difficult? And the answer is chromatin. The way the DNA is packaged, the way the genome in a terminally differentiated cell is packaged, is a barrier, albeit not an insurmountable one, to de-differentiation. So if we want to understand IPS and if we want to understand normal differentiation of stem cells, I argue that we have to understand chromatin. Okay, so now I've used this word enough times that I probably should have defined it already. Chromatin is simply the assemblage of protein, RNA on the genome. Okay, so it's a number of factors. It's the physiological form of the genome in a eukaryotic cell. It achieves this great compaction I alluded to, but it's also a key regulation point to accessing the underlying DNA. So the chromatin environment around a gene is a critical determinant of how easy it is to turn that gene on or off if it's already in one of those 2 states, or how readily changeable the expression pattern is. But it's also key to accessing DNA in other paradigms, like DNA repair, for example. So simplistically, chromatin can be thought of as sort of in 2 different states. Now, this is sort of in a local sense. You could have cells that have, largely speaking, heterochromatin, so this more condensed, compact chromatin. I'll show you a micrograph in a moment. And then you can have a more transcriptionally accessible euchromatin sort of state of a local patch of cells, okay, of nucleosomes, I should say. So what are these things? These are nucleosome core particles, which are sort of the fundamental repeating unit of chromatin. Essentially, it's an octamer of core histones, colored H2A, H2B, H3, and H4. So these are the 4 core histones. You have 2 copies of each of these histones per nucleosome core particle. You can think about this as really kind of a spool about which DNA is wrapped. So this is, at least in small part, a resolution to this compaction problem, but it doesn't explain how you get 6 orders of magnitude of compaction. It explains maybe a little bit of the compaction, right? You're wrapping DNA around a spool of protein. And so the higher order compaction of these nucleosomes is really where most of the actual compaction comes from, the higher order structure. Okay, so there are several epigenetic information carriers that have been implicated. The most classic and well-established case of an epigenetic information carrier is DNA methylation. Okay, so methylation of the cytosine nucleobase at the 5 position. It doesn't alter at all the Watson-Crick base pairing sense of C. What it does is it creates a sort of a binding platform for proteins. We'll talk about in a moment. The other putative epigenetic information carrier, so this is well established in the sense that there are mechanisms by which this can be maintained through replication in the semi-conservative sense we'll talk about. And then, so histone modifications represent another putative epigenetic information carrier. Certainly they modulate chromatin structure. The crux of the question is, are they heritable in a discrete sense? And then non-coding RNA species also play some role in this. Okay, so histone modifications are post-translational modifications of the histone amino acids. So enzymes come in and install additional chemical groups onto these amino acids. And in some way, these chemical groups encode information. Okay, so how do these encode information? How does DNA methylation encode information? What are the consequences of these things? We'll talk about in depth from here on out. First, I want to show you this beautiful, elegant structure of Tim Richmond's of the nucleosome core particle in 3 dimensions. So you can really see it's negative 1.8 superhelical turns of DNA wrapped around this core of histones and colored as described on the side. There's a tetramer along this dyad axis of symmetry about which the nucleosome is rotating. So the entrance and exit are sort of pseudo-symmetric in the sense that you can rotate it 180 degrees and see something that looks very similar. So it has this just beautiful, elegant symmetry to it. It's almost wedge-like shape of the histone octamer about which nearly 2 turns of DNA is wrapped. Okay? And the salient thing also to notice here are these tails. It's hanging off into space. These globular, out of the globular region of this folded core octamer are these histone tails, and that's where most of the action's occurring. That's where a lot of the histone modifications that I described, these post-translational marks of the histone proteins occur. Okay? Okay, and this is sort of the cartoon version of the compaction. So you go from this sort of duplex DNA to the 10 nanometer fiber to a 30 nanometer solenoid fiber to various higher order structures till you get down to a mitotic chromosome. And so this is really how you achieve compaction, these layers of structure. So you can imagine, right, if you're in a 30 nanometer fiber, this DNA is far less accessible than in this 10 nanometer fiber. So simplistically, you can think about the partitioning between sort of a closed down chromatin state to this higher order structure versus a sort of a euchromatic, more open state in this sort of 10 nanometer fiber that's much more accessible. The DNA is accessible to transcription, accessible to any sort of manipulation. So here's what beads on a string look like when you take an electron micrograph. Here's what this 30 nanometer solenoidal fiber looks like from a micrograph perspective and from a model perspective. And then sort of macroscopically, if you just look at a whole nucleus and you contrast stain it in electron microscopy, you can very clearly see sort of dark patches that represent heterochromatin and then lighter patches which represent euchromatin. This figure is a little bit misleading in the sense that it might lead you to conclude that euchromatin represents most of the genome. Actually, about 95% of the genome is heterochromatin, so that most of the DNA is in these darker, more condensed spots. Although in terms of spatial occupancy of the nucleus, most of it's euchromatin. So these more open states occupy more space within the nucleosome, consequentially. Okay, so again, this is sort of the simple dichotomy that I would be remiss if this wasn't one of the take-home lessons. Just sort of partitioning between heterochromatin and euchromatin is one of the local structure changes that sort of controls access. So to the transcriptional apparatus or to DNA repair, et cetera, or to the replication origin, for example. So you can easily go, well, from a heterochromatin state to a euchromatin state, just at a local level of, let's say, 4 nucleosomes or 3 nucleosomes, just by compaction. Okay? So how is this achieved? And how do histone modifications and DNA methylation play any role in this? And that will be sort of the bulk of what we talk about for the remainder of the talk. So what do chromatin modifications do? Well, simplistically, they help toggle this sort of path between heterochromatin and euchromatin, either directly, so the modification itself in some way impacts the ability to compact or the stability of a given nucleosome, or in an effector-mediated way. So a binding partner comes in and binds the histone modification or the DNA methylation or the epigenetic modification. And in some way, as a consequence of that binding, it recruits an activity that maybe opens up the chromatin or closes it down. So we'll talk about some examples of that in a moment. So I just want to underscore how incredibly subtle these modifications are. Right? So when we're talking about a post-translational modification of protein or a methylation event of DNA, we're talking about removing a proton. And I've just drawn a space-filling representation of what these things look like spatially. We're removing a proton and we're adding a methyl group, or removing a proton and adding an acetyl group. So these things are incredibly, incredibly subtle. Right? There's very little in between a proton, chemically, in between a proton and a methyl group that biology has access to. So this is about as subtle as it gets. Yet still, this subtle nuance of adding a methyl group or 2 methyl groups or 3 methyl groups to a lysine, for example, is sufficient to determine a whole signaling cascade. So it's just absolutely amazing that nature is capable of recognizing the difference between these things at such high fidelity that a signaling cascade can hinge upon it. So these things are small. Just to give you sort of a more sort of perhaps nuanced view of how subtle these effects are, I've drawn the nucleosome core particle here, again, the core histone octamers here, in a space-filling representation. And I'm going to turn on 2 methyl groups on the DNA and see if you can see them. It's pretty subtle, right? So now I'm going to turn them yellow so it's a little bit clearer. And now I'm going to circle them. Does everyone see them? Okay. So we're talking about adding 2 tiny bumps that don't change much in terms of charge on the DNA. They change nothing in the way of charge. They're just adding, substituting a proton for a methyl group. That's about as subtle as it gets. Yet still, there's a protein capable of making this discrimination. This is an example of a protein called MECP2 that specifically binds DNA when dimethylated at a CPG step, so-called CPG island, where both C's in a dinucleotide repeat are methylated at the 5 position. And contingent upon that methylation, this protein binds. Absent this methylation, there's a 1,000-fold weaker affinity of this protein for this piece of DNA. So just these methyl groups can toggle that affinity. And that 1,000-fold factor difference is sufficient to specifically recruit this complex to sites of methylation. Now, it is often the case that sites of methylation aren't just a single CPG step, but are groupings of CPG steps. So there's an avidity effect in the sense that you can have multiple substrates for this binding event on a small patch of DNA, so that if this binding event does sort of fall off, right, because it's being sampled very rapidly, its probability of rebinding another proximal methylation CPG step is higher. Okay? So by these means, it seems that biology has been able to actually localize a repressive complex to regions of DNA. And in particular, DNA methylation is always repressive. Okay? So it always recruits repressive activities to shut down transcription. Okay? So what does this do? Well, it turns out that this MECP2 complex acts on histones. So it deacetylates histones and actually recruits, so histone deacetylase activities, which is another post-translational modification of the protein, the histone proteins, that in ways that we'll get into is capable of also shutting down transcription. So this isn't acting directly on transcription. It's acting very indirectly. It's acting by altering the local histone modification profile. Okay. So just to be perfectly clear, I want to make sure that I'm not doing any prestidigitation here. I said earlier that nothing is changing about the capacity of DNA when we're talking about adding this epigenetic modification, this 5-methyl-C. And just to be clear, the base pairing capacity of 5-methyl-C is completely identical to normal C, right? So its capacity to code for a G, both during DNA replication and during transcription, right, of a message, it would code for a G in both cases. Okay, so we're not changing anything about the coding properties in the Watson-Crick phase. We're just adding this little bump that serves as a recognition platform. Okay, so as I said earlier, these things often occur in CPG islands. So you have an avidity effect that essentially increases the residence time of this repressive complex at a region typically a promoter, to silence it. So you can go from an active to a repressed version of the gene simply by adding methyl groups to these CpG islands. And so that's one mechanism by which transcription of genes that aren't necessary for a cell, for example, neuronal genes like some sort of or an olfactory receptor gene is silent in a muscle cell, right? The muscle cell shuts down transcription typically by this sort of mechanism. Okay, so here's the cartoon version. It's a little small, I apologize. Can you guys see this? Hopefully you have younger eyes. So here's sort of a scheme of how this could work in a sort of a mechanistic sense. You, in some way, and this is a very interesting question, recruit DNA methyltransferases to an active locus. So the DNA gets methylated. You can see it's already acetylated on histones. These little blue flags represent acetylation. And so this methylation in turn recruits this repressive complex that deacetylates the histones locally and installs additional histone methylations that recruit additional activities that shut down transcription, effectively changing the environment. There's also some additional chromatin remodeling typically as a consequence that's not depicted here. And so the reason why this is truly epigenetic, right, is that it's got the semi-conservative mechanism of inheritance. Much like DNA is semi-conservatively replicated, you can have, upon a round of replication, you get hemimethylated strand, which is the daughter strand, and the parental strand is fully methylated. There's a maintenance methyltransferase, DNMT1, that comes in and fills in hemimethylated species to recapitulate the fully methylated strand. This happens in S phase as well. So it's following right after a replication event. And so this is how DNA methylation is maintained through mitosis. It turns out that it's also, at least some of it is maintained through meiosis. So you can inherit your methylation patterns from your parents in interesting ways that hopefully we'll get a chance to talk a little bit about. But all this describes sort of a process by which methylation can be installed de novo and maintained. How are methylation marks removed? And so this area has actually been pretty controversial for some time. A number of labs have reported mechanisms that seem chemically improbable for removing DNA methylation marks. Recall, this is a carbon-carbon bond, so it's about as stable as you can get. It's very difficult chemically to activate a carbon-carbon bond. It's one of the holy grails of organic catalysis. So it's a difficult chemical problem. And so it turns out, recent papers suggest that there is indeed a pathway for removing this methyl group, but it's not direct. It involves, so it's sort of hard to see here, but this is a methyl group, which is sequentially oxidized by this TET family of oxidases. 1011 translocated is what it stands for. It's an oncogene family. It's hard to exactly understand how this is oncogenic, but in some way, this plays a role in several different cancers. In any case, the enzyme activity is to oxidize the 5 position sequentially from hydroxymethyl C or 5-HMC to 5-formyl C to 5-carboxyl C. And then it becomes a substrate for base excision repair, which is a set of enzymes that just cleave this glycosidic bond and allow repair polymerases to replace it with a C. Okay. So this is the pathway by which we now think that methyl groups are removed, a fairly recently discovered pathway, but clearly the most robust from the particulars of the enzymes and their activities and the genetics that underlie this. And there certainly are processes in the cell that involve massive loss of methyl groups. In particular, during fertilization of an egg, both paternal genomes are massively demethylated. Not entirely, but massively. About 90% of methylation is lost upon egg fertilization from both the paternal and maternal genomes. So which methyl groups survive is sort of an open question, and how are those the heritable ones is sort of the million-dollar question in this field. So I mentioned before, right, we have this sort of paradigm of a repressive activity arising as a consequence of a binding partner recognizing 5-methyl-C specifically. Well, if I tell you that the steady state of hydroxymethyl C is actually pretty appreciable in certain tissues, like brain, adult brain, it's about 0.7% of all C. So we wondered, since there's a high steady state level of this in the brain, and also in embryonic stem cells, oddly enough, we wondered, could this be a signaling molecule in its own right? Could it be sort of a unique epigenetic information pathway? Of course, it's derived from 5-methyl-C, but we wanted to know if there was an orthogonal signaling system that recognized hydroxymethyl-C or formal C to the exclusion of 5-methyl-C, right? So a pathway that's independent of methylation in the sense that it doesn't recognize methyl groups, this binding partner, this reader, if you will. Similarly, we know that the methyl binding partners actually don't recognize any of these intermediates. So we wondered if there was a new information pathway here to be discovered. And this would, of course, require a specific binding partner to transduce some activity downstream of some sort. We don't quite know what the outcome is in terms of function, but the hypothesis is, since this is a long-lived intermediate, and since things that bind this don't bind this, perhaps there are specific proteins that bind hydroxymethyl-C or these other oxidized C forms. And perhaps these binding partners can be discovered from a tissue source where this is relatively abundant. Okay, so it turns out this is very abundant in the adult brain. It accumulates as a function of age in ways that we don't fully understand. So we looked for a binding partner for 5-HMC in brain extracts. This involved getting a lot of pig brains. This is me and my graduate student in the cold room grinding up pig brains to make a nuclear extract. So biochemistry can be very, very visceral. We have essentially a way of going from intact brain to brain smoothie to isolate neuronal nuclear extract to the exclusion of glial cells, et cetera, and other support cells. So we get nuclei from neurons via this sort of fancy way of fractionating brains. And then from there, did sort of classical biochemistry. So we take this pig brain nuclear extract representing about 20 pig brains and run it over a series of columns. Have you run across column chromatography or fractionation biochemistry? Is it worth talking a little bit about how that works? Okay, a little bit. So the idea is if you have a very complex pool of proteins, how do you purify the one protein you care about? In this case, the one protein we care about is a protein that binds hydroxymethyl-C, right? No one's discovered it before, so how do we discover this protein? The point of this is to discover a factor and purify it, okay? So you run a series of steps to essentially break that protein pool, that's a very complex starting protein pool representing all the brain's neuronal proteins in the nuclei of a pig brain. And we fractionate it into smaller groups of proteins by running a series of columns. So there are ways of, essentially these are both ion exchange columns, but there are ways of sort of differentially adhering protein pools to columns based on their charge properties. That's what these first two steps do. And what we do is we collect fractions at each sort of individual step of this gradient, let's say, of this column. And each one of these fractions we then screen in an activity assay, okay? So here, activity assay is a gel shift assay. Let me explain how this works. We have a radio-labeled, so it's radioactive, just so we can see it. We have a radio-labeled hydroxymethyl-C free duplex, okay? So it's got symmetric hydroxymethyl-C at a CpG step in the middle of this duplex, and it's radio-labeled so we can see it. And then we just look for the formation of this complex by gel. So we incubate it with various fractions from these columns, so fraction 8, fraction 10, fraction 12, et cetera. And we look for the appearance of a complex. So the complex manifests itself in a gel as a band with lower mobility than the free DNA, okay? And actually, you can kind of see two activities here. There's another faint activity here at a different mobility. So the main activity we've been following is sort of in these fractions of this particular column. And you can see the protein is actually relatively pure at this point. There are only a few bands on a gel, which we've identified by mass spectrometry. And so the question you might ask is, okay, you have some binding of this oligo, but is it specific for hydroxymethyl-C? Oh, this is just a trace of a UV trace of the column chromatography, this last column chromatography step. And it just shows where the fractions are relative to where the absorbance of protein signal is, okay? So we do a specificity gel to measure essentially how specific is this binding activity. Is it specific for hydroxymethyl C? Does it also bind methyl C? If it also bound methyl C, it wouldn't be a very interesting information carrier in the sense that it wouldn't be uniquely recognizing any of these oxidized intermediates. And so we screen each of these oligos against sort of an equal molar concentration of methyl, hydroxymethyl, formal, carboxyl, and regular C. And only in the cases of hydroxymethyl C and formal C, so these two oxidized intermediates, do we see binding. With an equivalent amount, and in other gels, a factor of 100-fold more of methyl C and car C and regular C, we detect no binding with this activity. Again, suggesting that it's rather specific for hydroxymethyl C and formal C. Those of you who've had organic chemistry and know something about aldehydes might suspect that this is probably maybe not the right way to draw it in water. It's probably hydrated, so it looks sort of like two hydroxyls coming off of this carbon rather than the aldehyde moiety. Again, suggesting that that's probably the element of recognition that this activity that we've isolated binds. Okay, so we've actually isolated and purified a factor that's in some way involved in specifically binding these two nucleobases, these two novel DNA bases that exist in the human and pig, thankfully, genomes. So what are they doing? That's the million-dollar question. And that's what we're working on actively right now. We're trying to figure out what is the functional consequence of this protein binding event now that we've cloned the proteins. And we're working with them to recapitulate the activity in vitro. But we're excited because we feel like we have the first hint of a completely new epigenetic information pathway. I mean, it's pretty rare you get a chance to say that. Simply because, I mean, I started the lecture saying there are 3 putative epigenetic information carriers. Well, maybe now there's a 4th. So this is exciting. We're really interested in sort of following up and trying to understand the functional consequences of this very specific binding. The nice thing about, I should also point out, is that it doesn't bind 5-methyl-C and regular C, so it clearly is orthogonal, right, in the sense that this binding activity doesn't recognize the precursor nor the end product of this oxidation pathway. Again, suggesting that's rather specific for the intermediates. Okay, so enough about DNA methylation and its discontents. Now, what about histone modifications? Well, histone modifications are a considerably more diverse set of entities. There are approximately 250, at last count, identified possible modifications of histone proteins. And this is just of the core histone proteins. There are other histones that don't form this nucleosome core particle but attach out here, essentially, called linker histones, and they're also modified as well. So there's just this massive diversity of methylation, phosphorylation, acetylation, there's no way you could possibly read this, but if you want, it's from this Kouzarides paper. But this shows the enzyme that actually installs the modification, and in some cases the enzyme that removes the modification. So maybe a useful sort of cartoon level framework of thinking about this is histone modifications and epigenetic modifications more broadly have sort of 3 phases. They have a writing phase, an enzyme that installs them, an erasing phase, an enzyme that removes them, so that you can control the level of that modification in a locus-specific way. And then they have a reading phase, so the actual information transfer. So the presence of this modification contingent upon this presence of this modification at this particular site on this histone, let's say a methylation mark. So a reading module binds strongly contingent upon the modification and the flanking sequence. And so often it's this reading event that in turn transduces downstream function, recruits other factors that either open up chromatin or directly contribute to transcription or transcriptional repression. Okay, so just here's the same sort of game played with histone methylation, again to show that it's a very, very subtle thing. So I'm turning on one of the histones, 3 methyl groups. You all see it? Okay, I didn't need to do the usual trick. All the way up on the very tail of the end of the H3 tail, it's lysine 4 that's being trimethylated here. And that in turn recruits a number of complexes, one of which is this BPTF complex, which is a chromatin remodeling protein. So essentially, the presence of this histone modification in turn recruits this remodeling activity, which slides histone octamers around on DNA to reposition and reconfigure the local chromatin environment. So it, in effect, activates transcription by opening up a more dense region of chromatin that makes it more permissive for the transcriptional apparatus to dock. Okay, so how does H3K4 get there in the first place? So we talked a little bit about the consequence, and maybe we'll talk a little bit more about it in a moment. But I want to start by sort of now making a little bit of a connection to some more biomedically interesting phenomena. The writer of this mark, or one of the main writer of this mark, is called MLL1, which stands for mixed lineage leukemia. It's a gene that's frequently disrupted in leukemias, about on the order of 10%. I think the statistics are actually on this slide. Yeah, so cumulatively about 10% of all leukemias, if you sum AML and ALL, involve MLL translocations. So essentially, it's a gene lesion that occurs pretty regularly. And it occurs pretty regularly as a consequence of normal cancer chemotherapeutic treatment. There's a sensitive breakpoint in the MLL gene that when you dose someone with a drug like a toposide or VP16, cause essentially this chromosomal level breakage to occur. So we're basically taking the gene for MLL, which is a methyltransferase, and we're splitting it in two. It's also a major cause of very aggressive infant leukemias, most of them, in fact, which are completely untreatable. I should also point out that these are some of the most difficult to treat and also the most fatal of leukemias out there. So this is just an example of the different other sorts of lesions that give rise to leukemias. BCR-ABL is, of course, the lesion that's treated by the Gleevec kinase inhibitor. So this is one of the more successful examples of a targeted cancer chemotherapeutic. And so MLL rearrangements are extremely lethal and have very poor prognoses. Okay, so what is MLL doing in leukemia and in development more broadly? Well, it seems to be the key regulator of this very interesting set of clusters of genes called Hox clusters. Diagrammed here is the Mexican wrestler Santos, just to show you which body segment of Santos the various subsets of Hox genes are expressed in, okay? So in Santos's shoulders and head, this portion of the cluster is on. And in his midsection, this portion of the cluster is on. And in his posterior, this portion of the cluster is on. And this is really important for Santos's development early on, from when he was an embryo up to being an infant. These genes firing basically defined his segmental identity, okay? So this is an important sort of set of genes for determining segmental identity in all bilateral organisms, from fruit flies to man. And it turns out that, it sort of governs AP axis symmetry and body segmentation. So it turns out that this very same pathway is repurposed for hematopoiesis, the development of blood cells from a hematopoietic stem cell in your bone marrow, right? You've heard of bone marrow transplants. We're talking about transplanting hematopoietic stem cells, essentially, into patients and killing their old cohort that is ridden with leukemia. So leukemia is essentially a disease of incomplete differentiation. Typically what happens, you pick a particular lineage, either lymphoid or myeloid, and you end up being in one of these late stage precursor stages that doesn't fully differentiate. And it's capable of self-renewal and forms what's called a blast stage. And that is, in effect, leukemia. It's incomplete differentiation to a terminally differentiated blood cell, okay? So this very same HOXA cluster, in particular HOXA9, is critical for this last stage transition, and I'll show you in a moment a little bit more clearly, from a committed precursor to a final differentiated blood cell of some sort. Okay, just to give you a sense of the segment identity in a more sort of dramatic case, this is a HOX mutant called Antennapedia. So it's basically a mutation in the HOX gene. And what this does is gives you arms coming out of your face if you're a fruit fly. So again, underlying the fairly important aspect of these genes being expressed sort of spatially properly in a developing organism. Otherwise, you get really horrible things that happen. Okay, so how are HOX genes regulated? Well, it turns out they're regulated, largely speaking, by histone modifications rather than by traditional transcription factors themselves. The Hox genes themselves are homeotic, sort of pleiotropic transcription factors that control a number of genes involved in body segment identity. But how are the Hox genes themselves, in turn, regulated? Well, it turns out it's sort of this steel cage grudge match, if you will, between this activating histone modification, this K4 trimethyl that we spoke about earlier on histone H3, so H3, K4, lysine 4, trimethyl, just to give you the lingo one more time, and this K27 trimethylation event, which is installed by this sort of opposing methyltransferase complex called the polycomb repressive group complex number 2. And so you have these two complexes that both methylate lysines on the same histone tail, but have completely opposing activities. And so you can read out the chromatin state of a Hox A cluster, for example, and say which genes are on, which genes are off, just by looking at the patterns of methylation, K4 or K27 methylation. It seems to be the sole, one of the key indicators and drivers of gene expression of Hox genes. So the question is, how is one trimethylation of lysine activating and another one repressive? You need to have orthogonality in your reader systems, again, getting back to this concept of orthogonality. But here we have an even bigger problem. We have our modification being exactly the same, trimethyl lysine. So we have to recognize the flanking sequence context of the trimethyl lysine very, very exquisitely. Otherwise, you might inappropriately activate rather than repress, right? The downstream consequence. So how's this done? Well, here are two ribbon diagrams of structures that show reading events of trimethyl lysine. This is trimethyl lysine in the context of lysine 4, so you can see a flanking arginine 2 on the very end terminus of the histone tail, the histone H3 tail. And you can see this recognition is built into this aromatic cage. We can talk about that in some detail if you want later on. It's this fairly interesting recognition, non-covalent type of recognition, a bond called a cation pi type of interaction. Not in your standard textbooks these days, but maybe will be. Similarly, the same sort of aromatic caging is used by this polycomb chromodomain to recognize lysine 27. But instead it recognizes lysine 27 in a completely different way. So it recognizes the flanking residues. Both of them recognize the flanking residues and exclude K4 in this case, or K27 in this case. So here's maybe a slightly better way of looking at it, sort of a surface view. You can see it's a very shallow cleft on the surface of this PhD finger and that the histone peptide sort of slots into, whereas the recognition of K27 is a little bit more intimate. It actually forms a continuing portion of this beta sheet of the polycomb protein itself when it complexes with it. So the elements of recognition are substantially different apart from the aromatic cage. Okay, so enough structural detail. Just that's to convince you that we truly understand the structural and mechanistic underpinnings of how these two different readers work. So let's talk about the outcomes. All right, so we have methylation, which recruits this BPTF PhD finger. There's an additional modification which also helps recruit this BPTF protein. The BPTF protein is itself a chromatin remodeling protein. So what does that mean? It's in an ATP-dependent way. It actually opens up chromatin by sliding a nucleosome around on DNA. So sliding a histone octamer along DNA is maybe the better way of thinking about it, which in turn makes that DNA a little bit more open, that region of chromatin a little bit more open, and transcriptionally permissive, so that the pre-initiation complex of hollow polymerase can dock down and start making RNA. It turns out the very same modifications that are involved in recruiting the nerve complex are also involved in stabilizing basal transcription apparatus. So the basal general transcription factors have the capacity to bind these very same histone modifications, intriguingly. So we were curious mechanistically how this worked. Ignore some of the details of this figure, apart from we have purified nerve complex that we've made recombinantly in insect cells. And we have a remodeling assay where we look at a position nucleosome be repositioned sequentially into from the N1 position to the N2, N3 position in the presence of NERF and ATP. So this is what it looks like normally. You see, you're sort of starting largely in this position, and at 1 minute you've got a little bit of N2, and then as a function of time you see N3 populate and N2 populate more. Okay? So this is effectively chromatin remodeling at a single nucleosome level, right? If you cut off the histone H4 tail, you lose that remodeling entirely. Okay, so this is an interesting observation that I and my technician made. If you delete this tail, you lose remodeling activity. None of the other tails matter, but just this H4 tail. So Richard went on to look for a stable interaction between this histone tail, H4 tail, and this nerve complex, this remodeling complex. Okay, so we had a great positive control because we knew this PhD finger of BPTF bound H3K4 trimethyl, right? Because that's the structure I just showed. So, and we know the affinity of this. So we did basically a peptide pulldown assay where we immobilize the peptide on a bead and see if we can fish the complex down with the bead after washing a lot. So this is what our protein looks like to start with, and we enrich it by adding a bead that has this peptide attached to it. Although the unmodified, the unmethylated peptide doesn't retrieve any BPTF complex. And then we looked at a series of H4 peptides. So this H4 1 through 20 seems to also stably bind nerve somehow. And this H4 11 through 30 also seems to stably bind nerve in the sense that we pull it down in the presence of this peptide. Now, Richard wanted to design this GAGA mutation. So essentially taking the center overlapping region of H4 and changing it to, coincidentally, a glycine-alanine step, which he called the GAGA mutation. And it turns out this GAGA mutation completely kills the binding activity, suggesting that the binding interface is somewhere in this region, certainly involving some component of this KHRK, or KRHRK, sorry. So right now we've isolated this to a subunit of this complex, and indeed a portion of a subunit of this complex, and we're trying to grow crystals of it to try and understand structurally how this works. Then we're hoping to re-engineer a modified complex so that we can actually understand sort of the underpinnings of this recognition. Maybe we can make a nucleosome that will a complex that will remodel GAGA-containing histones, depending on what our structure tells us, if we can engineer it or not. And so no one's ever actually shown that the function of BPTF, the critical function of BPTF, it's an essential gene for development, but no one's ever shown that its essential function is actually chromatin remodeling. So we think that some of these accessory alterations, mutations we engineer from the structural work, should give better mutants to plug back into model organisms to show that's the remodeling function of this complex that matters as opposed to some other complex function. Okay, so I managed to sneak in 2 pieces of stuff that's going on in my lab. I still have quite a bit of time, I guess, don't I? Good, so we can talk about some more biomedical cases, which I have slides for here. So here's sort of a cartoon version of Hox gene cluster activation. Somehow the MLL complex is recruited, and I could talk a little bit more detail about an aspect of that my lab is working on in understanding how this is recruited by non-coding RNA. This complex installs histone modifications, so in particular H3K4, because it's a methyltransferase, but there's also an accompanying acetyltransferase activity that installs an acetylation on the H4 tail. This then recruits NERF, and NERF then remodels that region of the locus, making it more transcriptionally permissive. And the very same modifications contribute to the stability of the pre-initiation complex that starts actual transcription. And so you can imagine this actually as a sort of a feed-forward loop, particularly if some of the RNA that's being made is not messenger RNA, but is nearby non-coding RNA, which in turn recruits this, which is something that we've discovered to be the case for MLL1. So we're working on trying to understand this recruitment of MLL component of this process right now in the lab. So let's get back to cancer. So we sort of understand how it regularly works, how MLL activation works in a regular sense. So what happens when you have an MLL tandem duplication, MLL partial deletion, MLL translocation? And all of these have been isolated from leukemia patients. So you go from, we were talking about differentiation of a hematopoietic stem cell to a committed myeloid progenitor or committed lymphoid progenitor, right? So these are Bs and T cells. These are all sorts of other cells, including red blood cells. So how do we get to a macrophage? Well, you can follow this pathway down of differentiation from this hematopoietic stem cell to this committed myeloid progenitor to another sort of blast stage, sort of last level or penultimate level commitment of commitment in terms of progenitors. And so if you look at HOXA9 expression, it's on in all three of these cases, and then as a consequence of methylation at the HOXA9 gene, recruiting BPTF, recruiting polymerase to essentially potentiate transcription. And then if you look at the very terminal stage, HOXA9 is turned off by PRC2 activity dominating this H3K4 trimethylation. So H3K4 trimethylation has to be erased at HOXA9, and K27 trimethylation has to be installed in order to go from this stage to this stage. And so this is what happens when you have, these are K27 trimethylated. So what happens when you have an MLL fusion is you have dysregulation of the methylation. And inappropriately, this HOXA9 gene remains on and methylated. So the methylation patterns don't switch at the final stage of lineage commitment. And so what you get essentially is this blast stage, this last precursor stage, self-renewing indefinitely. Essentially, that is leukemia. These are leukemia cells. Essentially, they're cells that leave HOXA9 on, and many leukemias leave HOXA9 on, and inappropriately so. Okay, so that's essentially how our present understanding of sort of epigenetic regulation of HOXA9 works and plays with leukemia. I should point out that there's some MLL-targeted therapeutics that are not quite in the clinic yet, but people are actively working on trying to design drugs to target that process. It's a little bit complicated because I presented a slightly simpler version of reality, but bear with me. Okay, so I want to give you another really cool example of epigenetics in biomedicine. So present day sort of technology for the treatment of HIV infection is essentially a chronic administration of this cocktail of drugs, reverse transcriptase inhibitors predominantly, although some cocktails involve protease inhibitor, HIV protease inhibitors. So, I mean, you're taking gram quantities of these drugs every day for the rest of your life just to kill any virus that pops out of a latent immune cell, okay? So HIV is a retrovirus. It incorporates itself into the host genome, in particular into host immune cells, right? And so the reason why we're just sort of treating it as sort of a chronic disease as opposed to something that we can actually cure is because we have this latent population of immune cells that have HIV integrated into their genomes, but are not actually expressing any of the genes that reconstitute the virus. So how do we go from this phase of so in order to actually treat and cure a patient of HIV, we'd have to treat both populations of cells, the ones that are actively producing virus and the ones that have latent virus integrated into their genome, right? And of course, the ones that are actively producing the virus are problematic because they're creating more virus, which is in turn infecting more immune cells. So to cure it, you need to both continue administering the cocktail of drugs, but you also need to, in some way, make the latent virus pop out of the genome and become active again. So yeah, I think I mostly said all of this. So is there a way that we can manipulate the chromatin state of infected patients? In particular, a way that the integrated viral genomes could be manipulated so that they become no longer latent. They become active and are thus treatable. And the answer so far has been using this drug that's an HDAC inhibitor called SAHA. So histone deacetylase enzyme is an enzyme that removes acetyl marks on histones like this. And so HDACs actually effectively increase histone acetylation, sort of in a way that sort of pleiotropically opens up chromatin if you treat cells with HDACs long enough. And so treating HIV-infected cells with SAHA, actually even at the patient level, so you're treating the patient with a cocktail. That's what this baseline ART is. It's an anti-retroviral, anti-RT, reverse transcriptase cocktail. Standard care for HIV these days. And you additionally treat them with SAHA. You can actually get the viral load in their blood to go way up, essentially. These are just different patients. So essentially, this work, which just came out in the spring, suggests strongly that we can actually activate latent populations of virus and treat them through several courses to completely exterminate them at this point. They've done several courses of this now, the authors who did this work. And it seems like they're seeing absolutely no viral load in the patients a year out after treating them. So this may be working its way very soon into the clinic in terms of a way of actually curing HIV. I should also point out, right, HIV, taking this cocktail is completely impractical in sort of third world countries or for people who are poor. This costs a lot of money to take this cocktail every day. So curing HIV is substantially going to benefit humanity, even though we have sort of a palliative treatment right now. Curing it is much more important than sort of treating it. Here's another really cool example of epigenetics in an addiction paradigm, a drug addiction paradigm. In particular, a friend of mine, Ian Mays, works with cocaine-addicted mice. And so he administers cocaine directly to the mice's brains in a conditioned place preference mouse model. So essentially they have a cage where the mouse lives and has two chambers that are distinguishable to the mouse. One is checkered, one is white. And the mouse, each mouse has its own cage and has a cannula going into its brain. And when it's in one of the two chambers, it gets cocaine at some frequency delivered directly into its brain. Okay, and so if you train a mouse under that setting, and then no longer treat the mouse, no longer give it cocaine in the cocaine chamber, the mouse's propensity to spend time in that cocaine chamber versus the other chamber, just by randomly walking around, is called this conditioned place preference. So the percentage of time that the mouse spends in the cocaine chamber versus exploring the other chamber is sort of a model for how addicted that mouse is to cocaine. Okay, so using a control group, which is addicted, he knocked out a histone methyltransferase enzyme called G9A. It writes this particular histone modification on H3, a different repressive mark. And so knocking out this enzyme caused the mice to become effectively far more addicted. So their conditioned place preference score went up, approximately from 100%, so in the control group, to 150%. This is massive in terms of these sorts of experiments. Usually conditioned place preference scores are very subtly different, and you kind of have to run the experiment millions of times to get anything significant. This was very significant with a small number of animals. And so the G9A knockout works. The modification that G9A is involved in installing is reduced in the knockout. It's not complete because the way he did the knockout is not a complete animal. It's a targeted knockout in a particular brain region. And then he did the reverse experiment. We actually infected the brains with a G9A producing virus to overexpress G9A. So if you knock out G9A, which is this histone methyltransferase, you get an increased addiction potential. If you have too much G9A, or ectopically more G9A than you normally have, you get sort of a reduced interest in administering in spending time in the cocaine chamber. So it's sort of a really kind of a cool, simple example, and we don't know which genes we're talking about at all. This is just sort of global levels of this methylation mark. So clearly these are regulating genes that are responsible for the animal's response. But it tells you that histone methylation is a critical intermediary in things like drug addiction, in things like chronic stress paradigms, in things like animal models for depression. All of these things, histone modifications are lighting up as clearly distinct in the two cases. So here's an example where we're very far away from understanding the mechanism, but the observation, the descriptive observation, is very clear that G9A plays a pretty important role in addiction. Okay, maybe I'll open up the floor for questions. Well, actually, that's kind of a fun little scheme. So this is sort of the sort of fun part about being an epigenetics researcher is, you're taught at a fairly early stage that Darwin was right and Lamarck was wrong. Well, I mean, a lot of these sort of heritable aspects of epigenetic environmental influences on an organism that can be inherited to the offspring are very Lamarckian in a way, right? A giraffe extending its neck to reach a tree branch that's higher, and in some way giving that to its offspring. It's not completely out of the range of possibility. I don't think that's a good example, but there are clear examples of environmental factors and aging affecting phenotype in a heritable way. There's a classic cohort of two cohorts of Dutch and Norwegian patients whose either paternal grandfathers or fathers were exposed to a starvation event. And as a consequence of methylation patterns encoded in differential methylation patterns encoded as a consequence of starvation, their children have several-fold higher incidence of cardiovascular illness. So that's a great example of how environment can actually be heritably encoded in DNA methylation and passed on to offspring. So there are many other examples of this sort of thing, just hints of this sort of thing, and understanding this in a deep mechanistic way is going to be representing maybe the next 20 or 30 years of research. But I just wanted to show sort of Lamarck as he aged here into this older man for fun. So DNA methylations change over time as a function of TET oxidation, as a function of DNA replication, not fully reconstituting full methylation patterns, environmental inputs, nutrition, and aging, all play a role in DNA methylation patterns over the lifespan of an organism. So to what extent is this heritable? It's kind of terrifying. There are also a lot of in utero sort of nurture factors that also manifest as DNA and histone methylation. So in addition to there being cancers that are wholly due to epigenetic lesions, essentially a tumor suppressor being placed in a state of being repressed epigenetically, but having no actual genetic lesion. So there are cancers that are derived just from inappropriate silencing of tumor suppressor genes, for example. So I think I'd like to make the argument that chromatin and epigenetics is sort of the center of a lot of interesting biology and a lot of really interesting disease. And I hope maybe I failed if I presented you with the sense that we understand much of this at all. It's still a lot. There's still a lot that we need to work out. There's still a lot of mechanism that's missing. There's still a lot of really tantalizing observations that we don't fully understand. So still a lot of this work in this field is descriptive. So we'd like to understand these things at a deep mechanistic level and at a level that would enable generating therapeutics. Okay, so with that, I will hopefully try and end. This is my lab. This is Kate, the person who does the brain work. And then this is Richard right here, who does the chromatin remodeling work. And there's a lot of other stuff that I didn't get a chance to show. Any questions, comments, obnoxious statements? Yeah. What's that? Sorry, what was that?
I missed a few slides. Do you think you could put this PowerPoint on Moodle, which is our class?
Sure, yeah, absolutely. I think I gave it to one of the AV guys, but is that the act of putting it on? Okay, I certainly give you a copy, absolutely.
Also, you mentioned before that outside of just understanding the process of you like to re-engineer the actual proteins and kind of see how the structure plays in. Is that a common practice to re-engineer the sensitivity?
Well, it's not that common, but it's one of those things that is often the case. I mean, it depends on what you really mean by re-engineer. It's one of those sort of, my philosophy is that you don't really understand a system unless you can re-engineer it and get a predictable outcome, a changed outcome, whether that's in a living cell, living organism, or just even in vitro with reconstituted factors. So we're starting in vitro, but we'd like to go back into organisms and with some of these sort of re-engineered interfaces and see if we can change the targeting of NERF to a completely different constellation of modifications. You could certainly imagine just appending different binding modules onto the NERF polypeptide as opposed to this PhD finger and bromodomain might target it to completely different loci, and we could get completely different sort of expression patterns just by putting this weird, let's call it bizarro NERF in a cell or in an organism.
Like this isn't a method of isolation that everybody does.
It's not something that everyone does, no. Maybe it's something everyone should be doing. I don't know.
You mentioned the Dutch starvation when they tried to build the World War II, and they had impacts for several generations afterwards.
Yes.
I was curious, and maybe that epigenetics decisions by governments in the context of if kids get proper nutrition, I'm using a simplistic model,
Yeah.
it has actually implications for several generations later. Has any of this filtered up? Probably not.
Yeah, not that I know of. I mean, I don't know if you've been watching at least our government's level of scientific discourse, but it's embarrassingly agnostic of even the most fundamental scientific insights. It's almost sort of politics seems to be sort of antithetical to scientific findings these days. But you're right. I think it does. I think you're right. I think the fact that yeah, I mean, a lot of these aspects, I think it also could inform more effective neonatal care. I think these are the sorts of things that probably will be tracked in like the Star Trek future, but we're pretty far away. I mean, we're still at sort of the phenomenological level of understanding of these things. It's descriptive. The mechanism by which these methylations are deposited in the event of starvation, completely obscure. The methylation, the way these particular methylation events survive the de-imprinting that occurs upon zygote fertilization, completely unclear. And then how, I mean, I think the last component, like the genes themselves that are methylated and their role in cardiovascular health, there's some reasonably good explanations of those. But the first two steps are maybe the more interesting ones, and we don't really know anything about that. So yeah, I mean, it would be great to have epigenetics influence policy, maybe in the future. Not that I'm aware of right now.
I remember the slide where you mentioned that depending on the folding patterns of certain chromatids, so like whether or not they're in specific things, so whether or not they're linear or whether or not they're packed in more.
Right, so like a heterochromatin versus euchromatin, heterochromatin being sort of the more compact, yep.
I remember one of your applications was actually making it into a more linear kind of formation in order to increase expression.
Mm-hmm.
Have you looked at the possibility of perhaps looking at turning more chromatin back into the other, I guess, formation or expression?
Right. So toggling between heterochromatin and euchromatin and doing that in a controlled way, is that the question? I think that, so that would be really kind of the holy grail of sort of epigenetics therapy or engineering, if you will, to be able to, in a targeted way, to say, I want this gene off in this patient, and in some way get a chromatin remodeler or a histone modification pattern that signals for that chromatin remodeler to go to that particular site in the genome and close it down. So the simple thing to do, you don't actually even, you can do this completely agnostic of chromatin remodelers, but if you could in some way recruit specifically to that locus you wanted to shut down by turning it into heterochromatin from euchromatin, if you could recruit K9 methylation, like G9A, that methyltransferase, that methylation of K9 is sufficient to actually to segue between an open sort of euchromatin fiber to a heterochromatin fiber. So just that methylation signal alone, and you know there are a couple of factors that explain why that is, some of which is chromatin remodeling. There are other factors that just bind that modification and are dimeric. So they actually act as sort of just passive staples. They kind of bind one nucleosome's K9 trimethylation and then another one nearby or far away, and effectively passively staple two chromatin fibers together to make something more compact. So simply targeting K9 methylation would probably be sufficient to do that. Now, how do you do that in an engineered fashion? How do you get that into a human? It's just like a gene therapy question, but that would certainly be a way to do it.
How did you get started in your epigenetics career?
It's been a long path. I started off as a chemist. My major in college was chemistry, although I did almost a double major's worth of neuroscience. So I had an interest in biology, and I had a sense that that's where the interesting questions were. And that sense deepened when I went to graduate school in chemistry. So technically my PhD is in organic chemistry, but I did not so much organic chemistry when I was a graduate student. I chose a project that was more sort of biochemical and structural biological. because I got the sense that, I mean, there's still areas of chemistry that are interesting, but I get the sense that there aren't huge unknown problems. I mean, the question, I mean, arguably, if you want to talk about hypothesis-driven science, the hypothesis in organic chemistry is, or the question you're asking is, can I make that? And the answer is yes. It's just going to be how much suffering it is, like how many steps, how efficiently you make this molecule. But modern synthetic chemistry is advanced to the point where you can probably make that anything. People have made some incredible molecules, but that doesn't, I mean, that's interesting and useful for making pharmaceuticals, but it didn't strike me as a sort of big sort of discovery kind of thing. It didn't sort of light my fire in the way that discovering something fundamentally new about biology or something fundamentally new about, you know, yourself in a way, if you want to think about it, does. So I mean, I just think biology is a fantastic area where there are a lot of really sort of unexplored vistas and territories. And so, you know, I took the sort of mechanistic thinking from my chemical training, my chemical and biophysical training, and ventured further and further and further into biology. I still consider myself a biochemist, but we're doing, you know, omics stuff. We're doing cell biology in the lab now. So I definitely came to biology via a long circuitous path in the more physical sciences. And I think that's actually a pretty useful path in terms of understanding mechanisms at a deep level and thinking about problems quantitatively. So, I mean, even if you want to be a biologist, biology, as you read about it in your textbooks these days, is not going to be practiced at that level when you guys are actually in labs as graduate students, let's say, or undergraduates, or when you're running your own labs. It's going to be increasingly quantitative as a science, increasingly integrative, increasingly coupled to bioinformatics. So I would encourage you to look, if you're interested in biology, learn as much computer science, math, and physics as you can to equip yourself to be the biologist of the future.
Actually, I have a question about the role of SAHA in HIV. You stated it would draw out the actual copies of HIV, but wouldn't it still be in the genome even if it was drawn out?
Well, so the treatments not only inhibit HIV, but also kill the host cells. They're sufficient to kill the host cells. So, but that's a very good question. I mean, right? If it's not in every cell in the genome, it's just in the subset of lymphocytes. So, if you manage to target that entire population of lymphocytes, you cure the patient. But it's a very good question. Yeah.
So you were mentioning how possible treatment like marks, histone marks, and like accumulation and methylation of histone marks can result in, you were talking about latency of viruses, like HIV virus specifically. Do you think in viruses such as like the herpes virus, a methylation or accumulation of histone marks could change when the virus gets reactivated out of latency?
Yeah, so I mean that's exactly it. So maybe methylation therapeutics could work, much in the same way. So herpes and HIV sort of have similar life cycles at a sort of a gross sense, and so you still have to you still have to combat this sort of latent virus pool when you're trying to actually cure any sort of retroviral infection. And so, yeah, I mean, I think methylation inhibitors, I know that the group that did the SAHA work is actively working with a methyltransferase inhibitor that inhibits these repressive methyltransferase marks. It's called BIX, some number, I forgot, BIX 1029, I think. Not entirely sure that's correct, but there's a small molecule out there that targets K9 methylation. And so they're trying combinations of these two to get it more efficient, or just multiple courses of treatment. And their results so far have been pretty encouraging. This is a group at UNC.
What are some of the applications with
So I can tell you in my own lab, we're beginning to wrestle with some interesting and somewhat thorny computational problems. One is just dealing with massive data sets, okay? So when you do, so a lot of chromatin research, and I didn't really give you a good example of this today because I didn't want to spend 10 or 20 minutes explaining the technique, but a lot of chromatin research is somehow tied to next generation sequencing, which is a way of just sequencing tons and tons of DNA in very short reads, okay? So something on the order of 200 million very short reads of a fragment of genome can be shotgun reassembled and to reconstitute a whole genome. But the sequencing can also be used for counting statistics, okay? So if it's coupled to something else, so you can use sequencing not just to learn a genome of an organism, but also to do counting statistics. So if you want to know, most people, instead of doing RNA microarrays these days, are doing RNA-seq, so-called RNA-seq. Do you guys know about RNA microarrays, or is that? They're just ways of assessing relative expression levels of RNA using a chip. So rather than doing that, almost everyone has moved more towards just sequencing all of the RNA. So turning it, doing a cDNA synthesis and then sequencing it, so-called RNA-seq. And that generates massive amounts of data. The data sets tend to be something like 20 gigabytes worth of read data. So how do you process that in a meaningful way? Writing software or writing R programs or scripts to process the data is something that we're dealing with in my lab. There are other really interesting questions. There's a problem in my lab that involves the kinetics of spreading of a particular histone modification into a region. So going from heterochromatin to euchromatin, how quickly does that occur? Well, we have this reconstituted in vitro system for looking at chromatin spreading. And so we're trying to understand and model this data set of sort of sequential methylations and understand precisely how the enzyme that does this modification, how it moves around on the chromatin fiber. And it involves some fairly sophisticated modeling. But those are just two very selfish, self-serving examples of computation epigenetics. People are looking at histone modifications in DNA methylation genome-wide using sequencing types of methodologies. And again, how do you treat that data? How do you work with that data? It's a fundamentally computational problem. It does. So let me tell you an interesting story about X chromosomes. Flies do it completely differently, okay? So in flies, right, it's all about gene dosage of the X chromosome. In humans and metazoans or mammals, we inactivate, females, I should say, you inactivate one of your X chromosomes somewhat randomly and at a fairly late stage in development, right? That's why you get a calico cat with different sort of splotches of coat pattern coloring. It's a female cat, and arbitrarily the pigment gene for that particular cat, the dominant pigment gene, is X encoded. And so you get sort of different coloration patterns depending on how, which patch of skin has which gene, which X inactive. Okay, I digress. So is acetylation involved? Well, flies, acetylation is everything for dosage compensation. So flies do the opposite. Instead of inactivating one of the chromosomes, one of the X chromosomes, in males, they doubly activate the X chromosome, okay? There's a complex called the dosage compensation complex, which installs histone acetylation on lysine 14 of histone H4. Mouthful, doesn't matter, particular lysine acetylation. That upregulates all of the genes on the male cell's X chromosome by approximately a factor of 2. So, dosage and interestingly enough, that very same complex exists in humans and is doing something in X. It may also be upregulating X inactivation, but maybe not by the same extent. The active X. I'm glad you brought up the X inactivation because that's just a fascinating paradigm where a non-coding RNA or a series of non-coding RNAs ultimately recruit histone methyltransferases. And there very well could be an acetylation component to it in humans that hasn't been worked out yet. But so there's a non-coding RNA that just coats the inactive X called exist that recruits this PRC2 complex that I alluded to very briefly is the complex that installs lysine 27 methylation on histone H3, sort of the yin to MLL's yang. And so that's the way X inactivation works. You need an initial bolus of exist signaling to get PRC2 to that chromosome. Then PRC2 kind of sits around. It doesn't need the exist RNA to stay there. It just needs it to initiate. You can actually take away the exist at that point and keep that X chromosome inactive just because you've done the initial recruitment. But the stabilization of PRC2 is due to something else. We're still working that out, but really fascinating system. And yeah, acetylation could be involved in the other side.
Just a quick follow-up. How do you do that with just one X chromosome?
Yeah, well, it's really complicated. There's a series, I don't think I can actually even explain this competently. There's a series of like mutually transcriptionally interfering RNAs right near, there's like a series of, I think, 3 or 4 non-coding RNAs right near the exist promoter. And somehow, right, but how do you get switch-like behavior? How do you get a single choice? It's not entirely clear to me. So if you want to look, there's the person driving almost all of this research is Shirley Liu at the Dana-Farber. Actually, I think she's now just formally at Harvard Medical School. But she's done all this work, really fantastic work, trying to in some way explain the switch-like behavior. To be perfectly honest, I don't fully understand how the models of this sort of cycle of various non-coding RNAs influencing each other, ultimately influencing exist expression, is able to be that switch-like and to make a single choice per cell. It doesn't totally make sense to me, but they think they have it worked out. It's complicated is the short answer, though. It's a $1 million question.
But I think we need to finish up. Thank you very much.
Thanks for having me.